I think I'm also going to suggest that we wait maybe another 1 to 2 minutes in case there's some late arrivals. That's often the case for people to trickle in. Good to see everybody.
Likewise, Jessica. Happy Friday. It is Friday, I think.
Okay. All right. So again, thank you everybody for joining us. This is the second talk that is going to be given by Corbin Schuster. He is a candidate for the assistant professor of biology position here at Heritage University that would begin in the summer of 2022. Corbin already gave a wonderful lecture on Tuesday and was a teaching lecture in microbiology. And today we're very excited that he'll be sharing with us his research. So as before, we've instructed him to design a talk that would be appropriate for all walks of disciplines. Not everybody here is in the sciences. And that it be approximately 40 minutes long, so we would have time for discussion. Again, we are recording, and I will make the links available once I figure out how to do that. Corbin is from the Yakima Valley, where he graduated from Zilla High School in 2012. He also graduated from Heritage University with a BS in biomedical science under the direction of wonderful Dr. Gao. He then immediately began his doctorate in microbiology at Oregon State University, and he is scheduled to complete that degree by the end of the year, which is very exciting. His dissertation is focused on expanding diagnostic assays and characterizing the disease transmission of dynamics of pseudoloma neurophilia. And I am going to let Corbin take this away. I will say that the session is also being co-moderated, yes, sorry, the word, with Dr. Gao. And I do apologize, I'm gonna have to keep my screen off for the rest of the time, but Dr. Gao will be able to facilitate the question and answer session at the following Corbin's talk.
Awesome. Thank you so much, Jessica. I'll go ahead and share my screen real quick. Oh, it says host disabled participant screen sharing.
My bad, it's fixed now.
Okay. Cool enough. Okay. Hoping everyone can see that. I Ignash Maniksha Wakaikaboli Shiapochinki Corbin Schuster, Washnash Yakima Kanik, Na Alas Iwan Niksha, Klida Schuster, Ku Napusas Iwan Niksha, Elmer Schuster Sr., Skulikash, Oregon State University Pa, Kush Skuliana, Heritage University Pa. Hello everyone, my name is Corbin Schuster. I'm an enrolled member of the Yakima Nation. My grandparents are Klida and Elmer Schuster Sr., and I'm a current PhD candidate at Oregon State University, where I have been extensively studying. I guess my background is infectious disease and assay development. And so today I'll be talking about the expansion of diagnostic assays for pseudoloma neurophilia and characterization of the disease transmission dynamics. Before I get started though, I would like to do a land and people acknowledgement. Heritage University is on Yakima land and is the Yakamami Tikhcham is the land of the Yakima people. And so I'd like to give the adequate respect and land acknowledgement to that. And also, all of my research has been done at Oregon State University, which is on Kalapuya land. And so Oregon State University is a land-grant university and is located on the traditional territory of the Chepanefra Band of the Kalapuya. After the Kalapuya Treaty in 1855, the Kalapuya people were violently and forcibly removed and absorbed into what are now the Confederated Tribes of the Grand Ronde and the Confederated Tribes of the Siletz Indians. And so I'd like to give the adequate recognition to these three entities and nations in regards to allowing me to do research here at Oregon State University. Just a quick overview. I didn't know if students were going to be at this talk or not, so I wanted to give a little who is this guy, quick intro into my career path, and this will also orient us as to how I got interested in my research. And then I'll go over the background of comparative medicine and translational research, the history and impact, or the history of zebrafish and zebrafish as a biomedical model, the impacts of infectious diseases on this model, and then I'll get into the bulk of my dissertation. So my career path in STEM, it all started at Heritage. I was introduced to research by Dr. Nina Barcenas, where I believe I was a sophomore. And so I was introduced to the NSF molecular biology REU at Washington State University in the Dingler lab, where we studied plant diseases. From there, my passion in biomedical sciences really exploded. And I wanted to pursue a career path more in the biomedical realm. And so I was interested, I was interested in pursuing a internship program at the National Institutes of Health, where I studied correlations with failed dieting and satiety signaling in the brain. And from there, it really exposed me to the animal model in comparative medicine and using in translational research, what I'll get into here shortly. And then from Heritage University, I moved directly into my PhD. I got extremely interested in pursuing animal health and translational research in comparative medicine. And so I was funded by the NIH. And while I've been here at OSU, I've also participated in the OSU COVID Trace as a volunteer scientist. And I have, interestingly, my mentor from the NIH when I was there as an undergrad asked if I would be a core collaborator on this other COVID-19 project, in which we were looking at the race and socioeconomic status associated with chemosensory loss. So your loss of smell and taste as a key symptom of COVID-19. And then moving forward in January, I will do a short postdoc with the Zebrafish International Resource Center, which is also funded under the comparative medicine lens. All right, so moving into the research. So comparative medicine is a distinctive discipline of experimental medicine that uses animal models of human and animal diseases in translational and biomedical research. So the concept of this all came about of this one medicine, which emerged in the 1970s and eventually gave rise to the present One Health initiative, which essentially aims to unite human and veterinary medicine. And so through this lens, I was able to really identify, or I am able to identify with my research. And so translational research includes really two areas of translation. One is the process of applying discoveries generated during research in the laboratory, and then to the development of trials and studies in humans. But the second portion of that is translation that concerns research aimed at enhancing the adoption of best practices in the community and in the lab, and how we are able to report our findings using animal models. And so the animal models that we use are mice models. I focus with fish models, but there's also in the realm non-human primate models, dogs and cats, what have you. But what I'm going to be talking about today within the comparative medicine lens is specifically disease discovery and animal model development. And so there's several fish species used in biomedical research. There's, I focus on zebrafish, so we're definitely gonna be talking about that today, but there's also guppies, trout, and stickleback. All of these are fantastic host pathogen models that we can use to make inferences in regards to human medicine. But everyone always asks, what is so special about a zebrafish? Why should we, as a community, as a research community, invest so much into such a in such a model? And so one of the reasons for this is that there is a fully sequenced genome. There's we find that the zebrafish have a 70% similarity to humans, and more importantly, in regards to a host pathogen model, 82% of genes that correspond to humans also have a counterpart in zebrafish, and therefore we're able to make these inferences in these in translational research to sort of pose questions and solutions in regards to things that may be undoable or not, I guess, not viable in regards to human trials. One of the reasons also is that there's a high fecundity and high growth rate, and so these fish reproduce at a massive rate, and so we're able to have big populations in a quick turnaround time. They're also relatively low cost in maintenance. Of course, the husbandry personnel here at Oregon State University wouldn't say that, but they they are relatively low cost in regards to other models, such as non-human primates or even mice. They are completely transparent in embryonic development and rapid growth. Therefore, we're able to track disease progression through time, through real time. As you can see here on the right, I have this figure that's showing the different developmental stages of the developing zebrafish model. And so we're able to actually inoculate fish as soon as this time point here, and actually track that infection throughout the development and the growth of the actual of the organism. These fish also have an extremely adaptive nature. Their temperature range is anywhere from 4 degrees Celsius up to 40 degrees Celsius. And why this is important is because the average normal human body temperature is about 37 degrees Celsius. So we can actually inoculate these fish with human pathogens. And SARS-CoV-2 is a perfect model for that, in that we can actually inoculate these fish with this pathogen, and it actually recapitulates the pathology that we see in humans. And so the usage of the zebrafish model has greatly expanded, like explosive. And so over the past 20 years, the usage of the zebrafish model, and specifically models or papers that use the model in their research for publication, has greatly expanded over the past 20 years. And the outlook for this is looking like it's more and more of a premier model, because we can actually control a lot of factors within the environment that may be confounding in other settings. And so really the usage of the biomedical model has expanded to nearly every realm of biology. And so it's utilized in studies for psychology and neurobehavioral research, as well as COVID-19 and other infectious diseases, as well as cancers. And so it's important to realize that the extremely adaptive nature of this fish allows us to use it as a premier translational or the premier model for translational research. Where my expertise comes in all of this is really is the discovery of infectious diseases and the characterization of those diseases that impact this model, and therefore impact other research endpoints for labs and groups that are utilizing zebrafish in their research. And so there's three main organisms that impact this model. It's bacteria, parasites, and viruses. The bacteria that impact this model is Mycobacterium species, which is pretty ubiquitous in the environment, and therefore it causes a major concern in regards to confounding results in our research, in our translational research, but also background infections of this gram-negative septicemia species like Edsuella, or opportunistic pathogens such as Aeromonas. But where my expertise comes in is actually evaluating these parasites. And so my background is definitely within parasitology, within the infectious disease umbrella. And so today we'll be talking about Pseudoloma nerophilia, which is a microsporidian parasite that impacts the zebrafish model pretty significantly. There's also Plastophora, which is another microsporidium parasite, just not as common, and Pseudocapillaria tomentosa, which is capillary nematode, which is a little worm that lives in the intestine. And then there's a myxidium, which is a myxozoa. Myxozoan parasites are often can often be found in salmonid pathogens, such as Ceratinova shasta. And so this is a huge concern in regards to disease surveillance on the Columbia River or in the Snake River or any of the systems that contain salmonid fishes. And then, of course, there's the viruses. And zebrafish are susceptible to several. There's a lot of work going on this right now. I'm not necessarily going to touch on the zebrafish viruses, but these are the most common that we're seeing right now. And so we, I guess we as a research collective have examined over 15,000 fish from 2000 to 2017, before I even started here at OSU, but over 15,000 fish were examined and from 130 different laboratories across the country. And what we found was that Pseudoloma neurophilia was the most prevalent pathogen in all of the research facilities that are zebrafish research facilities. And so with this, we found that this high prevalence may have a significant impact to the actual model, right? And specifically because it's found in over 50% of all research laboratories. We'll get into it a little bit more, but what's important to note here is why that's so important is because most of the time that you won't even know that your populations are infected with the parasite. They will be completely asymptomatic, such as what we see with SARS-CoV-2, but spreading and transmitting the virus, or no, sorry, not the virus, parasite, getting mixed up. But we don't even know sometimes that this parasite is impacting our populations, and therefore that is a confounding result, especially in research that's utilizing this model. Specifically, what's coming to mind right now is for cancer. We work with this intestinal cancer in our lab, and it oftentimes, if we didn't know that pseudoloma was there, it could be a causative agent. It could be, contributing to the overall pathology and disease state that the state is, or that the fish is exhibiting. And therefore, it's just it creates all of these different variables that we would like to sort of exterminate from the worry, because it's a zebrafish facility. So the thing about zebrafish is that it's a confined system, whereas a river system is not a confined system. You have different flows, you have different temperatures, different pH, all these different variables that you cannot control. But in the zebrafish facility, now that's different. We can control all of that. We can control flow, we can control temperature, pH, and we can keep salinity, everything all at the optimal levels, so that we could actually analyze this without these different variables impacting our endpoint conclusions. And so microsporidium parasites are opportunistic pathogens. And so in humans are usually found amongst those who are immunocompromised, often HIV or AIDS patients. And these are obligate intracellular parasites. So they're immobile other than one of their defining characteristics. I'll get into in just a sec, but they're protozoan, so they're closely related to fungi, therefore eukaryotic, and there's a broad range of hosts. So some of them are host specific, some of them aren't host specific. Pseudoloma neurophilia is not host specific. And so we actually see pseudoloma being able to infect all kinds of different fish pathogen or different fish species, goldfish, guppies, swordtails. So they have a very broad host range. And they infect nearly all walks of life. And so there's lots and lots of fish hosts. And so lots of opportunity for new discovery of new microsporidium parasites. Some of the defining characteristics of this, though, is really this hardy resistant spore. And so the spore has a thick wall that with an exospore and an endospore layers that are outside of this plasma membrane. So if you will, the black layer here is the plasma membrane, and this really thick portion here that's in the gray is that double layer, which essentially confines all of the contents of that parasite within that spore, and it makes it to where it's resistant to chemicals, disinfectant chemicals. It makes it resistant to heat shock, pH, things, just common disinfectant protocols. And so one of the coolest things about this parasite is this polar filament here. So this is a really tightly wound filament that essentially, once the parasite it gets excited or it wants to move to a different host cell, it'll actually extrude this entire polar filament to move. So this is the only apparatus in which the parasite is able to be mobile. And so taxonomy is the branch of science concerned with the classifications of organisms. And so there's hundreds of species of microsporidia, and several genera infect different fish. As I mentioned earlier, I guess I got ahead of myself. Most are host specific, but not pseudomonas, as we know. And so how we are able to identify these is through the identification of the spore morphology and these different developmental stages, as well as comparing this the ribosomal DNA. So this is just a really quick image to show the evolutionary relationships of these different fish microsporidia. This was provided by my advisor, Dr. Justin Sanders. And I just wanted to show this to sort of to show that I don't know what's going on, to show that the microsporidian, the Pseudolomoneurophilia, is actually evolutionarily related to the salmonid microsporidia that we know. And so is just a very cool connection that I like to point out, that is not necessarily clustering with the other zebrafish microsporidia. And so, as I mentioned, one of the coolest portions and the more defining characteristics of this parasite is its ability to actually extrude this polar filament. And so considerable research has been directed towards determining the mechanism of spore activation that leads to polar filament discharge and the transfer of the sporoplasm and subsequent host cell infection. And so the initial observation about this process was the microsporidian spores swell prior to polar filament discharge. Decades later, another group came in with various concentrations of peroxides and salts and decreased the rate of filament discharge. So based off of these two studies combined, the hypothesis from the community was that these studies, that the filament discharge resulted from being actively researched, but the extrusion of the polar filament and the polar tube is what makes the life cycle of this parasite continue. And so how this operates is it penetrates the host cell in some species, and the sporoplasm is completely pushed through. So if you will, in this right-hand corner, I have this demographic of a spore, and it shoots the polar tube out, and through that, it pushes sporoplasm. Essentially, it's like an egg, essentially, in which it's going to push into the next host cell, and that's the only way that the parasite can disseminate throughout the body. And here I have an actual microscopy picture of that spore excitement. And so Pseudolomoneurophilia spore excitement occurs. We're able to artificially stimulate this spore excitement. And how we did that was through a chitinous spore and just simple UV fluorescence. And so the fluorescence will actually activate some of the spore germination, and they'll just in this case, it's not coordinated, but in the body, it's very coordinated. And so we were just here trying to show that the polar tube will extrude upon excitement. And as you can see, it extrudes 200 to 300 times its own size and shoots that sporoplasm all the way through to the next host cell. So just really fascinating machinery in order to infect. And so the general life cycle of this parasite is that there's three, there's a question. There's three major phases to the parasite. There's the water off-host infectious stage. There's which is wherein the spore is free in the environment, and it's very infectious and very resistant to disruption, heat, what have you. And then once the parasite moves intracellularly, so once the polar tube is extruded and the sporoplasm makes its way into this neighboring host cell or into a host cell, the parasite then moves into a proliferative phase, which is characterized by a growth phase of, or I guess a replicative phase of morogeny, which is a form of sexual reproduction where a parasite replicates its own nucleus inside its host cell and then induces cell segmentation. This then moves this proliferative phase into a sporgonic phase. And sporogony is characterized by the spore formation of these parasites. So once it is a full viable spore, then it can move, it oftentimes is spread through feces or urine, or just simply some fish die within a population. And so other fish will eat on that other fish through cannibalism. And so there's all these different modes of transmission in regards to the parasite. I think it's important to understand how the parasite disseminates through the body. This is an active field of research, but right now the way it works is we understand the spores are ingested through the gastrointestinal tract, and then they make their way, they penetrate through the intestinal wall and infect nearly every single organ system before making its way into immunoprivileged areas such as the central nervous system, where the parasite actually conjugates and it forms aggregates of spores within the immune system. Now we have to understand what that means, is that why it's moving into the immune system, or why it's moving into immunoprivileged areas such as the central nervous system. The central nervous system isn't going, you're not going to want all kinds of inflammation within your within the central nervous system, within your brain, and because that'll just cause all kinds of problems down the road. And so, I'm sorry, is there a question? Oh, okay. Anyways, so you don't want, so where they find protection is within the central nervous system, because there's not going to be inflammatory cells that are going to cause this to attack this parasite, right? And so it finds some protection within the central nervous system, and we'll get into the actual dynamics of this because it's very cool here shortly.
Hey, Tammy.
So transmission occurs both horizontally and vertically. Horizontal transmission means that the parasite is transmitted from fish to fish, so just being housed within the same tank, but it's also vertically transmitted. So it's transmitted from mother to progeny or offspring. And this occurs in two routes. It occurs intra-ovum, so inside the developing embryo, and it occurs extra-ovum, so in the ovarian fluids. And what's very cool about this in our hypothesis was that spawning events is a huge event for transmission. Anyways, but what's very cool about this is here on the right-hand corner is an actual developing embryo of the zebrafish. And these opaque plaques are actually aggregates of spores, tons and tons of spores. And so if you were to squish that out onto a microscope slide and actually look at it under the microscope, you'll see thousands, and I'm not exaggerating at all. Thousands of spores, up to 20,000 per parasites per fish. And so you can get an absurd amount of parasite from a single fish. And so you can imagine how this will impact the growth and development of this organism. There's also behavioral impacts, and there's so many studies on the behavior of this parasite in the fish. So I'm not going to get too far into this. I just wanted to summarize it all for everyone. Was that there's altered startle responses. There's a subclinical fish avoid capture more than infected fish. This was a unique research finding. There's also this incredible increase to stress and anxiety, which is in regards to disease and disease progression. It's crucial to understand and mitigate these problems. There's reduced activity and sex-specific impacts to exploration. So we see these two go together really, is that the females oftentimes are less active and less willing to explore with their tank mates. Where it gets really fascinating and where my mind just goes is the pseudoloma neurophilia actually alters brain transcripts. And so there's it's associated with 4 biological pathways which are enriched, associated with immune function. And these there's 14 gene ontology terms enriched that are also associated with immune function and circadian rhythm. So this is a heat map of the top 40 differentially expressed genes that I was specifically talking about, the different brain transcripts that were downregulated, in the biological context of an organism's production of gene projects, their downregulation is the project or the process by which a cell decreases the quantity of cellular components, in this case RNA or protein, in a response to an external stimulus. So specifically in this case, the parasite is causing a downregulation of key genes that allow the immune system to function and clear out any pathogens within the body system. And so this is suggesting a novel method for immune suppression and immune evasion. So these disease mechanisms are quite fascinating in regards to how the parasite is able to proliferate and essentially continue to transmit. And so I'm sorry, just fair warning, just getting really immunological on y'all. But the key function of the immune system is to locate, isolate, and exterminate foreign invaders or infectious pathogens. And so in this case, the parasite is actively working to silence the expression of the antigen presentation, which is critical for identifying and exterminating the pathogen from the body. And so here it is the MHC1 and MHC class 2 molecules that are being impacted, the significant impacts here. And so these molecules will present antigen to your immune cells to actually say, oh, hey, there's this pathogen here. You need to recognize it. You need to get it the heck out of here. It's causing all this different pathology. And so here, this heat map is really showing that, is that, if you will, forgive me, but there's 40 different differentially expressed genes here, and these are they all have relations to the immune system. And so here in the blue, and this red bar indicates that all of these fish are control fish. They're otherwise healthy as to what we call healthy. These fish here are all in the treatment group. So they've all been infected with pseudoloma. And so the sections of the brain that we were actually interested in looking at was the hypothalamus, the brainstem, the optic tectum, and the telecephalon. And so it's just fascinating to see this extreme different transcriptome based off of this parasite's infection. And so concern in the research community erupted over these findings of infected fish, to where they were saying parasitic infections may have spoiled zebrafish experiments. And so this has caused this non-protocol induced variation, which causes all these different confounding results, which has big implementation or big concern for people's endpoint research results. Pseudoloma is, like I mentioned, extremely resistant to disinfection. So even our common disinfectant protocols is around 25 parts per million, but there's survival even at 200 parts per million with chlorine. So these spores are extremely resistant. So therefore control is difficult as there's no treatment, and the parasite is extremely resistant to any of these disinfectant techniques. So this really gets into the nuts and cranny of my dissertation, is these diagnostic assays. In medicine, diagnostics refers to the process of identifying a disease, condition, or injury of an individual from clinical signs and symptoms. And in the case of histopathology, the diagnosis of the disease, occurs through analyzing host tissues under a microscope. And then for molecular diagnostics, in our environment, we shed DNA every second. So we are constantly shedding DNA into our environment. That is the same with every living organism. Thus, taking advantage of this fact, we can use genomic technologies to detect these pathogens within their hosts and within their host environments. The polymerase chain reaction, PCR, is the technique in which we use to go about doing this. And this technology is used for quick and easy amplification of DNA sequences, which is based on the principle of replication of these these nucleic acids. And so for pseudoloma neurophilia, previous work has already fully sequenced the genome of this parasite. So we can use a portion of the ribosomal DNA to detect the parasite with molecular diagnostics. And so we are specifically assessing whether the present is absent or is present or absent. So the phenotypes, as I mentioned earlier, so your population will be largely asymptomatic. And then these these other phenotypes that we see, this emaciation and the skeletal deformities, are not necessarily pathognomonic for this pathogen specifically. Therefore, like this pathology is not specific for microsporidosis, is what I'm saying. And so so this is a huge concern if you don't even know that your populations are infected. And so histopathology, we can use histopathology to actually locate these different parasites. And so here in panel A, this I actually did this panel myself. We actually see these spore aggregates starting to form within the spinal cord. And so using histology, we're able to actually say, to verify with the molecular diagnostics, okay, so we found the parasite in the environment. Okay, and then with histology, we can go through and validate, okay, we also find the parasite within the host, therefore it's really there, and we're not just seeing this anomaly in regards to observation. So getting into the molecular diagnostics, we have, we found that there's already a developed molecular qPCR assay that looks specifically at the central nervous system tissue. And so you need to pull out the spinal cord and hindbrain and able to use this assay. And so this group here, this is Marcus Krim, or Dr. Marcus Krim. He was looking at, okay, can we take that assay, that same exact assay, and just look at environmental samples? What he found was that it largely failed. This is another heat map that just shows that there's no real good detection here with pseudoloma neurophilia in regards to environmental sampling. This was consistent with another group who looked at the Miller et al. Paper, who they were trying to compare different molecular diagnostics and comparing that with environmental samples of all these different fish pathogens. So here, if you direct your attention to this graph, the P nerve is here. And so they found that the parasite was highly abundant in the fish tissues, but they couldn't find it in the water. And so we beg the question, what gives here? Why? And so that's really where my research comes in, is we believe that these spores need to be disrupted in order to release free DNA into the environment, right? So they have a hardy resistant spore that's resistant to disinfectant to heat, and all these different things. And we have to remember that nucleus, where the DNA is actually confined within that parasite, is confined tightly, bound around all that polar filament and all those different cell walls and different plasma membranes. And so within that, we believe that we needed to disrupt that spore, release some of that DNA into the environment, then we would actually be able to detect it. And so that was the first part of my dissertation, is the development of environmental assay for pseudoloma neurophilia. So we wanted to look at environmental samples. We believe that environmental samples contain inherent inhibitors for molecular diagnostics. Thus, environmental samples need to be analyzed using techniques that will also minimize inhibition. And so our hypothesis was that transition to this new advanced PCR platform would allow, in combination with sonication, which is a method for disruption that I'll talk about just shortly, will allow us to detect the parasite in the water, as well as these sampling methods will impact detection probability. And so digital PCR allows for a more consistent detection of pseudoloma neurophilia. And so this is different than traditional PCR in that it uses an absolute quantification. And how it does this is it obtains precise and accurate measurements by partitioning each sample into thousands of nanodroplet discrete measurements. And instead of the traditional PCR methods would take one measurement and just analyze that based off fluorescence, in which we would have to attain a standard curve in order to confidently say, okay, these samples are infected at whatever rate. Whereas with this is absolute quantification. So based off of these partitions, these individual reactions, which look specifically for the target marker within every reaction, we're able to say that there's different concentrations based off of these negative and positive ratios. And so here, that's what I'm showing here, is that here you have your negative samples, Up here in the green, you have your partition, your positive partitions. And based off of this, we get that ratio. We can say, oh, hey, okay, so there's only 50 positive partitions. This is a really low concentration. Obviously, the parasite is not very abundant. But if we got 15,000 partitions, then we could say with confidence, oh, hey, this sample is extremely contaminated or is extremely prevalent with this pathogen. And then we can make inferences based off of that. So our first objective was development and validation of a water test, which was extracting these brain parasites. And so it's a little cumbersome, but we essentially pulled out all these from about 80 to 100 fish. We pulled out the spinal cord and the hindbrain. We don't need the forebrain, just the hindbrain where the parasite aggregates. And we homogenize that to actually purify the spores in water. The second step was to optimize the spore disruption that I was talking about. And so that the first step our initial thoughts based off previous work was that the spores need to be physically disrupted in order to release DNA into the environment. So how sonication works is that it applies sound energy to agitate particles in liquid. And so using this machine, this Diagenode Pico bioraptor, we actually can place our samples in the machine, and we can send sound waves throughout the water bath that will disrupt those spores and release DNA. Well, our hypothesis was correct. So we were able to detect, using these different sonication techniques with these different time limits, we were able to actually detect the parasite using these PCR assays and at these different time intervals. So we were interested in finding that optimal spore sonication time in which the DNA was released from the actual organism. And so moving forward, we took those purified spores that we made from extracting the spinal cords and hindbrains. We took that and we wanted to actually see what our limit of detection for our assays were. And so how we did this was we spiked water samples. So we took negative control tank water and we put parasite and inoculated it with different concentrations of parasite. We sampled that through, we took 2 1-liter samples and filtered that through a 0.45 milligauss filter. And then we did the sonication DNA extractions and subsequent digital PCR. Filter is a very specialized membrane filter that allows us to dissolve it without harming dissolve in acetone without harming the DNA content. So what we found was that we had a detection limit down to 77.5 spores per liter, which was fantastic because the reported infectious dose is 300 spores per liter. Therefore, we were well below that threshold. Here, let me explain my graphs. We have spore concentration on the x-axis, and then on the y-axis, we have our log copy numbers. This is essentially the log copy numbers are looking at how many copies of this gene did we find within this sample. And so we used 2 serial dilution series. We used a series of 100,000 down to 100 spores per liter, and then we used another one of 77,500 spores per liter down to 7.75, and we found consistent detection down to 77.5. I know it can be a little confusing for filter A and B, but these are technical replicates. And so we took, these are the 2 1-liter samples and we filtered 1 liter through per filter. So therefore, we just wanted to make sure that our results were reproducible. Objective 2 was deployment of a water assay in OSU zebrafish facilities. And so we sampled 5 tanks over a 3-month period. And this, we were interested in determining the diagnostic application and determining if what we see in the environment is reflective of infections seen in fish individually. And so to do this, we set up 5 tanks of known infected fish in populations of 30. And we sampled them over a 3-month period. We sampled water, filtered it, and did the subsequent DNA extractions. But what's important is that we also were interested in looking at different sampling techniques, right? How does this impact detection? And so we incorporated treatments into our analysis to determine detection differences based on between flow samples, static samples, and spawn samples. And how we defined this was our flow through systems our general fish facility is a flow through system. So you have water coming in and constantly going out. So how this works is we thought that, okay, so if water is constantly going in and parasites being shed by the parasite, then it's also being constantly taken out. So we wanted to examine what happens if we left it static. So we left our tank static, no flow for 8 hours. And then we also, at the end of the day, we would come back and then take our water sample. And then for the no flow static spawn, we left that static overnight because, as we mentioned, that the transmission occurs both horizontally and vertically from mother to progeny. So we wanted to actually analyze, okay, are spawning events a huge route of transmission? What we found was that static sampling provides the best detection. There's different sampling methods that yield different results. And so we found that static tanks here, excuse me, I'm sorry. The static tanks were most telling for diagnostics. Detection was also strongest under static conditions. So here we have our different tanks. We have our percent positive by tank sample. So this just means how many of the water samples were actually positive for detection. And then this is the validation that we were actually looking at tissue specific. So the fish within every tank. And so as you can see, the water samples were almost at every time point much more telling in regards to diagnostics, in regards to what we were seeing in the environment. And then, of course, this just highlights that the detection is strongest under our static conditions. Even static spawn showed a great detection rate for pseudoloma nerphilia, although we couldn't quite answer whether spawning events is a huge route for transmission. So to complement the trends we were observing with our assay, we implemented a multi-state occupancy model to evaluate trends and biases in parasite detection. So the traditional models typically predict, but our model was a model of probability. So we were just wanting to confirm what we were seeing with our observations. And oftentimes these models are much more conservative than our observation. And so this multi-state occupancy model evaluates trends and biases in parasite detection, specifically evaluating relationships between habitat, sampling method, distribution, abundance, and detection of parasites in the environment. And so the occupancy detection parameters were used to estimate the number of samples required to accurately determine the detection of punionophilia in a tank under each treatment, and places each count with each replicate under 3 categories: not detected, detected but not abundant, and detected and abundant. I think it's important to talk about the traditional use of this occupancy model. It was used commonly in cell-mounted fisheries and have focused on detection and non-detection for detection probability estimations. However, the multi-state occupancy model here is estimated in consideration of the 3 states that I listed here. And so very cool was my modeling work, and the occupancy model revealed key differences in detection with relation to sampling method. So it essentially validated what we saw with our observation, and I wasn't just reporting some anomaly. And so here in the gray bars, we have the parasite when the parasite is present but not abundant. And in the black bars, we have when the parasite is present and abundant. And when the parasite is abundant in the environment, we have very good probability of detection. But when it's not, we don't necessarily have that. And so, but what's important to consider here is that under every condition that the abundant, when the parasite is actually abundant, we get really good detection. The diagnostic application is consistent with the occupancy model. And so the this is just really re-emphasizing that our model and our observation, they correlate, and that we see this percent positive by sampling method. Our observation was about 80%. Probability of detection with the parasite when the parasite is actually abundant was 94%. And so detecting this parasite when it is abundant, we felt confident. It's important. I know everyone like their eyes shoot over to the 64% because that's actually a low detection rate. But what's important to realize here, and it's the same with what we see with SARS-CoV-2, the rapid antigen test, is that when you have a population that you can consistently sample, you can increase that sensitivity, right? You can increase the sensitivity because you can constantly sample out of that same population. But when you are testing a vulnerable population, or a population that you know you're only going to be able to see once and test once, then you need something that's more, around this 94% detection rate. But the 64% detection rate isn't bad because we are in confined systems. We can serial sample even after the first initial sampling. And so we can increase this detection rate up to quite a bit with serial sampling and just ensuring doing the test over and over. Unsurprisingly, we found that the detection in water increases with prevalence in fish. And so also consistent with the occupancy model is that it appears that detection in water increases with fish prevalence, thus better detections when the parasite is present in abundance. And so our significant findings for this, I only went over one portion of the actual of my dissertation. I'm still writing the second portion of my dissertation. But the significant findings of the AIM-1 was that the detection that we were able to detect down to 77.5 spores per liter. With this, you have significant difference with sampling methods in regards to how you sample the water. And so we found that the static sampling is actually, static sampling is most telling for diagnostics. And then the increase in prevalence in tissues amongst the population results in increased prevalence in the environment. And very cool is this is the first report of integration of the occupancy model for zebrafish laboratories. I should have done it, but I didn't make a slide. Our future directions here would to be directly to integrate occupancy modeling and expand these diagnostic panels into zebrafish facilities so that they can eradicate this parasite from their facilities. Simply, unfortunately, it's not through a treatment or control. It would be through euthanization of infected populations. At this time, I'm happy to take questions.
Yeah, thank you, Corbin, for your talk. And I think our colleague, Dr. Miller, has a question. Yeah, Dr. Miller.
Okay, think outside the box question. By the way, fantastic work. Incredible.
Thank you. Thank you.
Incredible work. So you have a really nasty cell wall on this spore. We know of antibiotics that go after cell walls, peptidoglycan synthesis. What approaches have been taken to go after the structural integrity of the cell wall or the filament integrity? That's the other thing too. If you could attack the integrity of the filament at the molecular level, then when they try to inject it, it's not successful because you have a defective filament. So would any of the common strategies used for affecting cell wall synthesis or filament synthesis in other microbes be effective on the spores here?
Yes, actually there is. So our group is talking about starting an actual experiment for treatment. And there's this notion, I like how you brought up the filament, is the peroxidases. We see that these, the peroxides somehow impact this osmotic nature that we see with the integrity of that spore. And so we see that it diminishes that want, or that filament extrusion. And so I personally think that going down the route of looking into this peroxidase, or looking into the peroxides, and then looking into temperature, I think temperature will be a thing. And I think that's a simple I guess innovative or integrative strategy that we can use, that, it's the first, I guess the lowest hanging fruit that we can do that no one's actually examined yet, is just
See, that's huge, Corbin, because peroxidases are enzymes. And just like we at Pfizer and Moderna use mRNA as the template for making proteins to go after the spike protein of the coronavirus, you can make plasmids that you can introduce into the water. Those plasmids would be absorbed by the fish. And then as they're absorbed into the cells, be able to make the peroxidases that could inhibit. So I'm trying to think of what could you introduce to the stream that would help the fish fight off an infection of the drug. And so peroxidase, that's huge, because that could be practical in saving zebrafish life. So,
Oh, absolutely. Absolutely.
okay, good stuff.
Thank you.
Great. Yeah, thank you, Dr. Miller, for the questions. Yeah, so we'll give a few moments. Jeff has a question.
Hi, Dr. Schuster. That was a really great, really informative presentation. I was wondering if you could talk a little bit more about the pathogen's ability to sense changes in osmolarity, right, as a function of the tube mechanism.
Right.
Because that seems like it must be able to sense an incredibly fine change in osmolarity, right?
Yes, absolutely. And it's I wish I could really expand on this, but it's a really active realm of research right now. Is we believe that there's proteins, right? There's spore proteins that are core to this parasite. And so understanding how those proteins are expressed and how that influences its ability to germinate that spore and to excite and extrude that polar filament is an active realm of research. But yeah, I totally, it has to have some sort of sort of mechanism in which it's able to tell to know. And I guess from a laboratory point of view, I could talk about how this how the parasite seems to conjugate. It's really weird, because we'll put it on a microscope slide and they're really sticky like structures. And so what we see is that they'll actually be attracted to, like if you have an air bubble in your cover slip, they'll actually be attracted to the edge of that air bubble. And it's really fascinating.
It's very cool. Thank you, Dr. Schuster.
Yes, thank you. Thank you, Melvin.
Great. Yeah, yeah, so these are all important questions. Any additional questions for Corbin?
Thank you, everyone.
Okay, so we'll give a round of applause. Again, thank you again, Corbin, for sharing really exciting really breakthroughs in microbiology research. Again, have a safe and healthy Friday, also upcoming weekend. Again, nice to see our HU colleagues and friends. Have a safe and healthy weekend. Yeah.
Absolutely. Thank you for having me, everyone.